So the mitochondria and the chloroplasts are the organelles that were brought into the eukaryotic cell through that process of endosymbiosis. Mitochondria are usually called the powerhouse of the cell. We find mitochondria in all eukaryotes. So we find them in animals, fungi, protists, and plants. People tend to forget that plants have mitochondria because plants also have chloroplasts. Okay. This is where we are going to see the site of aerobic respiration. This is how ATP is going to be produced most effectively in eukaryotic organisms. Again, um, the structure of the mitochondria is closely related to its function. If we did not have this particular structure, we wouldn't be able to do the function. So let's take a minute to look at this. We remember through that process of endosymbiosis, we ended up with the plasma membrane from that original prokaryotic cell, but then we also got some plasma membrane from that host cell that engulfed them and brought them in. Okay, so we have two phospholipid bilayers. We are going to call we have the outer membrane that originated from our larger host cell, the inner membrane, which originated from that ancestral prokaryotic cell billions of years ago, right? So now we call this the inner membrane. That means there's a space in between those two membranes. So we'll call that the intermembrane space. It's the space between the two membranes. And then we have the matrix, which is the very most inner part. It's the interior of the inner membrane. Okay. Um, uh, Mitochondria also have their own DNA, and that is a hangover from that prokaryotic ancestor. They also have their own ribosomes. They reproduce on their own, so they're not, the nucleus does not tell the mitochondria when it's time to split and make more mitochondria. The mitochondria can sense that for themselves based on the demand for ATP. Okay. Again, when we get to cell respiration, we're going to look at the structure of these mitochondria again and really dig into the details of that. But just, like let's lay the groundwork now. Okay, the chloroplasts are the other organelle that we got from the second endosymbiosis event, right? So the first one gave us mitochondria, and all eukaryotes have mitochondria. The second, right, then we had that a lineage that had another endosymbiotic event that gave us algae and then land plants. Okay. So this chloroplast, um, they're another cell-like thing, right? They have two phospholipid bilayers, and then they have these thylakoid straps, which are thylakoid stacks, which are also bound by plasma membranes. Um, and we'll get into all the details of the structure, but again, these guys have their own ribosomes. They have their own DNA. They have multiple membranes. So we know that there's going to be transfery things happening here. We know that these things are derived from prokaryotic ancestors and have been incorporated in part of this new eukaryotic cell. There are actually other kinds of plastids. So chloroplasts are one kind of plastid. Plants actually have lots of plastids. Leucoplasts are plastids that don't have pigments, right? So that's the thing that separates them from chloroplasts. Chloroplasts have chlorophyll, and then leucoplasts are the same, are plastids that don't have that photosynthetic capability. Okay. Again, they contain their own DNA, they have their own ribosomes, they're going to reproduce on their own without any information or direction from the nuclear envelope. They can either store or synthesize biomolecules. So there's kind of the ones that we see the most that we like are called amnioplasts. These are the starch storage containers. We tend to see these in plant roots. We'll actually probably look at some of these in lab when we get later on in the semester, and we can actually see the amnioplasts when we stain them with iodine. Okay. The alleloplasts store lipids, the proteinoplasts store proteins. We can have specialized amnioplasts that are storing those carbohydrates become stagoliths, which are going to allow the plant to sense gravity. So in the roots of plants, the stagoliths will settle to the bottom of the plant cell, and so then they know that that weighs down, and that's what allows plants. It'll change how their hormones are distributed, so that when they run into an obstacle, boop, they can grow sideways, um, and then know which way is down once they get past that obstacle in the soil. The same thing with your plants, that so that if there's a limited light source, the plants are going to grow toward the light, and then you have to rotate the plant, and then they're going to grow back toward the light. That this is one of those mechanisms. It changes the hormone distribution within the plant um, to know which way's up and which way's down.